Hello learners. As you know, in science subject, practicals are very important because if you are not habitual of doing practical work, you cannot utilize the knowledge which you have earned through the theory classes. So today we will discuss about some of the zoological practicals. In zoology practicals, spotting is very important component. In spotting, students are supposed to comment on spots which are displayed on the table. Usually 5 to 8 spots are given to the students and the allotted time for each spot is about 3 minutes. But any student can write comment on the spot only if he is aware of the structure of that particular slide which is displayed in the microscope. So what a student is supposed to do? First of all, he should write the spot number, then should study the slide very carefully in high power of the microscope. He should identify it, and after identification, he should write the classification of that particular specimen if it is a protozoan, then characters of the specimen, any economical importance of that particular species, if it is pathogenic or non-pathogenic, what is the common name of the species, and the allotted time is only 3 minutes. So in this short span of time, only a student can write the comments when he know too much about the specimen. So today we will study some of the slides, and from the protozoans we will study the first slide, that is paramecium. After studying the slide of paramecium, I will describe how to write the comments on it. First of all, you come to the structure of the animalcule. As you know, it is a unicellular animal. Unicellular means the body is not divided any cell, and it is microscopic. The structure is having the outer covering which is known as pellicle. This is the pellicle, which is the outer covering of the animal, and it is formed of protein. This pellicle is permeable, so oxygen can be dissolved from outer water inside the body of animal, or CO2 come outside in the surrounding water. Pellicle is having hexagonal plates which are not visible in this slide, which is under the compound microscope. But if you will study this slide in electron microscope, only then some hexagonal plates of the protein level will be seen, which are forming the pellicle. And due to the chemical nature of the protein, it is flexible, so slightly change the shape of the animal. Now the common name of this particular animal is sleeper animalcule, because it looks like the sleeper. The outer boundary is like the sleeper, so it is known as sleeper animalcule. Now inside the pellicle, it is protoplasm, which is having two parts, ectoplasm, endoplasm, and in endoplasm the nuclei are present. First of all, the ectoplasm. Below the pellicle there is a thin layer which is known as ectoplasm. It is liquid-like structure, and the inner part of the protoplasm, which is granular, which is known as endoplasm. In the endoplasm, two nuclei are visible. These are known as macronuclei and micronuclei. Usually the cells are having only one nucleus, but in paramecium two nuclei are present. This macronuclei, which is of larger size, this is controlling the vegetative activity of this animal, while the micronuclei, which is smaller, It is having the genetic material and mainly it is responsible for the reproduction. So now we will see the structures which are present upon the pellicle. These are known as cilia. Large number of cilia are present, and due to presence of cilia, the animal is known as ciliate. The cilia, these are the locomotory organ of the paramecium. The cilia are always moving alternately, and they are having two type of strokes. These strokes are known as effective stroke and recovery stroke. By the movement of the cilia, the animal is moving in anterior direction. So the cilia, they are the locomotory organ of the paramecium. These are also formed of protein, and all the cilia, they are having a connectivity by myoneem, which is not visible in this particular slide. Only in electron microscope you can see the myoneem or the myofiber which is present below the pellicle. And due to presence of this myoneem or myofibers, all the cilia, they are moving in coordinated manner. Now, this part is known as cytostome. Cytostome is invaginated part of the pellicle, and it is working like the mouth of the animal. When a water current is formed by the cilia which are present in the cytostome, a smaller food particle will be engulfed in this region and a food vacuole is formed. This food vacuole, they are working as the digestive system of the animal, and strangely, after formation of this food vacuole, they are circulating to all the parts of the body. At the time of movement, these food vacuoles, they are changing their pH. Initially, it is acidic in nature, and then it is basic in nature, and during the journey of this food vacuole, the food atom is digested. After the digestion, it is assimilated by the protoplasm. The food particles which are digested, they will move from this food vacuole into the protoplasm. So all the part of the protoplasm will get the nutritive substance. Then this food particle will be attached to any part of the pellicle, and suddenly when it bursts, the undigested food material will move outside the body. So this is how the food vacuole is working, and it is the digestive system of an animal. Now, two important structures are there which are known as contractile vacuoles. These vacuoles are for the osmoregulation. Due to osmosis, as the protoplasm is dense in comparison to the outer water, the various salts, they are entering, or the water is moving from outside into the body, and this water quality may disturb the concentration of the protoplasm. Then it is very necessary that this water should be thrown outside from the body of animal to the outside. So these contractile vacuoles are working. Initially, some tubules are formed. These tubules will collect the water from the protoplasm, and then it will be transported to the central part. Now the central vacuole is formed, and this contractile vacuole will move from one part to another part and will collect the water inside the protoplasm. Finally, this contractile vacuole will come to the pellicle, and due to contraction of this contractile vacuole, all the water inside this vacuole will be thrown outside from the body. So this is how the paramecium is controlling the osmotic pressure of the body. Now, this part, macronuclei and micronuclei. Macronuclei, this is of larger size. micronuclei is smaller and it is responsible for the control of reproduction. The genetic material is present in both macro and micronuclei. There are some characteristic features of the paramecium as they are very sensitive to the temperature, to the touch, to chemical contents. That is why they are known as negative phototaxis, chemotaxis, tectotectes, because if you disturb the water, the paramecium will move away from the point of contact. If some chemical is present in the water, again the paramecium will move away from the chemical. This is their method of protection. So they are very sensitive to the chemical, to the touch and to the temperature. One more very important point in paramecium, there are two types of strain present. Some which are having the kappa particle, then they are known as plus strain, and another they are known as minus strain. So plus strain and minus strain, two type of parameciums are present. So in general, this is the structure of the paramecium. But if you will study the slide and the allotted time is 2 or 3 minutes, so it will not be possible for you to write all these characters. Then how to write the comments on paramecium? The heading should be, first of all, you should write the name of the slide. It means it is the pole mount of paramecium. So this is the identification. Then you write the classification, phylum, class, order and genus. If you are aware, you can write about the why it is under protozoan as it is a unicellular animal. Then why it is in class Ciliata, cilia are present, and then the genus and the species. The species is not required at the time of identification. Now you write about the comments of the animal. The one is habit and habitat. So it is an aquatic animal living in ponds, ditches, pools or slow flowing rivers water. They are in abundance in the slow running water, but in river, if in mainstream you will try to find out, it will not be possible to get the collection of the paramecium. So this is the habit and habitat. Now come to the structure, commonly known as sleeper animalcule due to shape of the animal. The body covering is pellicle. Protoplasm is divided into two parts, ectoplasm and endoplasm. For osmoregulation, the contractile vacuole is there. Nutrition is holozoic. Holozoic means animal-like. For respiration, the pellicle is used. Then for reproduction, if the conditions are favorable, it will divide into two parts. This is known as binary fission, and macro-micronuclei will be divided into two parts. All the cytoplasm will follow the division and animal will be divided into two parts. Then it is a asexual reproduction. But if the conditions are not favorable, then this paramecium will reproduce by the method of conjugation. This name conjugation is very important because two strains are used in it. So this is how you can write the comments on paramecium. Keep in mind that 3 minutes are given for commenting, and you should select the comments which are very important, which are having the distinguished features of this slide. So this is the first slide, paramecium. Now we can shift to the second slide. Again, this is a slide of a protozoan which is known as Euglena. Now the pattern is the same. You have first to identify the name of the specimen, that is Euglena, then classify it, protozoa, flagellata, dinoflagellata, then the name is Euglena. Then habit and habitat of the animal. Euglena, again, it is a freshwater animalcule, unicellular, and first of all you study the shape of the animal. This is spindle shaped, somewhat blunt at the anterior end and pointed at the posterior end. So it is microscopic, unicellular, spindle shaped animal, usually living in a group, found in muddy water because it is using the chloroplast for the nutrition. Now the very distinguished features of this Euglena is presence of this flagella. Flagella, this is originated from the basic part of this cytostome. Here two points are there which are known as blepharoplast, and two microflagella are originating, and jointly they are forming the large size flagella. Due to movement of the flagella, this Euglena is capable of moving, so its movement is known as flagellar movement. As the flagella is single, it is also having two types of the stroke. One is known as effective stroke when it is pushing water from anterior to posterior, and second is known as recovery stroke when the flagella is coming to its original place. As the flagella is only on one side, so whenever the effective stroke is used, the animal is moving to the anterior direction with some tilting to the right side if the flagella is present on the left side. If flagella is present on the right side, then it will be tilted to the left side. So a screw-like movement will be there in the Euglena when it is using the flagella simultaneously for effective stroke and recovery stroke. Now the outer covering of the animal, this is known as pellicle. Pellicle is formed of protein. This is having the elasticity, and due to presence of elasticity, the Euglena can change its shape. Initially it is spindle shaped, but if the temperature is not suitable or the sufficient light is not there, it can shrink itself due to effective use of the proteins which are present in the pellicle. The inner part, this is protoplasm. Again, it is divisible into two parts, the ectoplasm and the endoplasm. Ectoplasm is always the without granules and the dilute liquid-like structure, while the endoplasm, it is dense and having the granules. And in this protoplasm, nuclei are present. Nucleus is the main part of this animal which is controlling all the activity, physical or the reproductive activity of the animal. So nucleus is the main part which is controlling all the activity of the animal. Now first of all you should study this anterior part of the animal which is known as gullet. It is comparable with the mouth of higher animals, though it is not a mouth. Now inside this part, when the food is engulfed to the anterior part, a food vacuole is formed in this region inside the protoplasm. Food vacuole, they are moving from one part to another part. This is known as cyclosis. Cyclosis means the movement of the food vacuole inside the protoplasm, and during this movement, again the pH of this vacuole is changing from acidic to the basic. So it can be compared with the digestive system of the higher animals, when in stomach the medium is acidic due to presence of HCl, and when it is coming up to the region of intestine, it is basic as the acid is neutralized. So the same is the pattern inside the Euglena. Initially the medium is acidic and slightly then it is neutralized, then basic, and again we should come to the back slide. So, the movement of this vacuole, this is for the digestion. Now, very important point or very significant feature of this euglena is presence of this eye spot or the stigma. Stigma or eye spot, this is the sensitive part of the animal, and this animal always tend to keep this eye spot toward the direction of the light. The stigma or eye spot can be compared with the photoreceptive organs of the higher animals, and the presence of eye spot is very abnormal feature of protozoan. Usually this type of structure is not present in most of the protozoans, but the presence of eye spot or stigma very significant. This is photosensitive part. The second important feature of this animal is presence of this chloroplast. Presence of chlorophyll is the characteristic feature of the plant, and with the help of chlorophyll, the plants are utilizing the chlorophyll for the synthesis of the food, that is photosynthesis is conducted inside the plants. But euglena is the animal which is having the chloroplast, and sometime it is placed by some of the scientists between the plants and the animal. Why they are putting it in under the plant? Because it is having the chloroplast and the nutrition is same like the plants. And by the zoologists, they are considering this animal because it is having this eye spot and all the other structures which are comparable to the protozoans. So, when this euglena is having the favorable conditions, it is dividing itself into the two parts by longitudinal binary fission, and this fission method is the reproductive method of the animal. A very remarkable feature of euglena is if the conditions are not favorable or there is scarcity of the water and the temperature is high, then the animal want to protect itself. A covering around the euglena will be formed. That stage is known as pamela stage, and this pamela stage is a cyst-like structure. In that condition, the euglena can survive for a long time in the muddy region also. So, this is the protective method of euglena, or we can say it's a adaptation by euglena to cover the body by a layer and avoid the condition of drought. So, whenever the favorable condition will again available to the animal, the cyst will be dissolved and the animal will be divided into 4 or 5 parts or daughter cells, and each daughter cell will form a euglena. So, this is the general comment of the euglena. But again, when you are attempting or going for the spotting, you will get less time and you cannot write in such a detail about the structure of the euglena. So, what will be the important comments for euglena? Number 1, this is the whole mount of euglena. Then habit and habitat, it is aquatic animal living in stagnant or the slightly flowing water in streams, ponds. Generally, the body is spindle shaped, covering is known as pellicle, protoplasm is divided into 2 parts, ectoplasm and the endoplasm. 2 nuclei are there to control the activity of the animals. For the locomotion, it is having the flagella, and flagella is used by effective stroke and the recovering stroke. Presence of eye spot, which is for the sensation of the light. Presence of chloroplast for the photosynthesis or the autotrophic nutrition, and pamela stage is there to avoid the adverse condition, and this pamela stage is adaptation. So, this you can write about the euglena. In the last, you have to draw the figure well level diagram. So, if you have studied the two slides, one paramecium, second euglena, and you know the detailed structure of paramecium and euglena, now you have to select which comments are important. And in the limited time, how to select the comments. So, more important characteristic feature should preferably be written. In the case of euglena, what are the features important? Presence of white spot, presence of chloroplast, presence of flagella, and the condition which is known as palmillar stage. To curtail the time, you should draw the well level diagram of the smaller size, and the color diagram is not required for that. So, then classify the animal, phylum, class, order, genus, species is not required. These are the two specimens, paramecium and euglena. Now you come to the third slide, which is amoeba. Amoeba, again, this is very common example. Now, how to comment on amoeba? First of all, you should see the structure of amoeba. It's a unicellular microscopic animal not having a specific shape. Whenever it is in a living condition, always it is changing its shape due to formation of these structures which are known as pseudopodia. These are the temporary structures which are formed on the body part of the animal, and due to presence of this pseudopodia, they are always changing its shape. The genus is amoeba, the species is Proteus. Now first, the body covering. This is pellicle. It is formed of the protein which is having the elasticity, and that is why it can change the shape of the animal. The protoplasm is there inside the pellicle. It is again divisible into two parts, the ectoplasm which is on the periphery or near to the pellicle, and the endoplasm which is dense and having the granule part. The nucleus is of large size, single nuclei is there, and this nuclei is the controlling factor of the unicellular animal, and this is controlling all the vegetative as well as the reproductive activity of this animal, amoeba Proteus. Now, what are these pseudopodia? Pseudopodia, these are the temporary structures, or you can say the enlargement of the body part. How they are formed? When ectoplasm and endoplasm, they are changing the density, so these projections are formed. In this particular slide, this is the food material. Whenever is amoeba, if it is moving near to the food, it will try to cover the food material from all the sides, and so these two projections are formed. One which is visible from dorsal side of this food material, and this is on lower part of the food material. By development of this pseudopodia, the amoeba will cover this food particle from all the side, and finally this food material will be engulfed inside the protoplasm, and it will be covered in a vacuole which is known as food vacuole. So, this type of nutrition is known as holozoic type of nutrition. What is holozoic type of nutrition? When we compare it with the higher animals, ingestion, that is getting of food material, then digestion of the food material, absorption of the food material, and egestion of the food material, that is throwing the undigested food material outside the body. So, just compare this, how it is engulfing the food material by surrounding the food from all the side with the help of this pseudopodia, and finally a food vacuole is formed which engulf food. So, this is ingestion. Then we will compare the digestion. This food vacuole is moving inside the protoplasm By the method of cyclosis, it is moving to all the directions, and during the movement the medium is changed. Basically or initially it will be acidic, then basic, then the neutral, and during the journey of this food vacuole, all the food material which was inside the vacuole will be digested and dissolved, then it will be absorbed by this protoplasm. So, all the part of the protoplasm will get the nutrients. So, we can compare this activity with the circulatory system of the higher animals. As you know, in higher animals, this is the responsibility of the circulatory system to distribute the digested food to all the parts of the cell. So, here this duty is performed by this food vacuole, because it is moving in all the direction and the absorbed food will be distributed inside the protoplasm. When this food vacuole is empty, it will come to the near to the surface. When it will be in touch with the pellicle, suddenly it will be burst and all the undigested food material will be thrown outside from the body. So, the digestive part will be completed. For respiration, how this all the three animals, how they are respiring? As you know, all the animals continuously require oxygen, and when this oxygen is utilized for the metabolic activities inside the protoplasm, the CO2 is formed, and this CO2 cannot be accumulated inside the body. So, it is to be thrown outside from the body, and when this amoeba, which is living inside the aquatic medium, the permeability of this pellicle allow oxygen to move from outside medium, that is water, into the protoplasm. Inside the protoplasm, this oxygen will be utilized for metabolic activity, CO2 will be formed, and CO2 will be permeable from this pellicle and it will move away from the body, that is the shifting of CO2 from the protoplasm to the water medium. So, that is how the animal is getting respiration. So, all these three animals, when they are living in aquatic medium, the process of respiration is almost same. Through pellicle they are getting the oxygen, and through pellicle the same they are transferring the CO2 from the protoplasm inside the water medium. So, how to write the comments on amoeba? Number one, first of all you have to identify the slide. This is the slide. In bracket you can write whole mount, amoeba proteus, genus and the species. Then the comments. First of all, the habit and habitat. This is an aquatic animal found in the stagnant water and in running water, but slowly running water. Unicellular, microscopic. Body is not having the specific shape. That is why the name of the species was originated Proteus. Its name of goddess who is changing the shape. That is why the name is amoeba proteus. The outer part is pellicle, inner part is protoplasm, divisible into two parts, ectoplasm, endoplasm, a nucleus of large size, controlling the vegetative and the reproductive activity of the animal. Food vacuoles are for the digestion. Pseudopodia are for the locomotion, and the method of reproduction is by binary fission. Sometime you may get the slides which is showing the amoeba which is having the stages of the division. Usually when the conditions are favorable, the amoeba can reproduce by two methods. One by binary fission, another may be by multiple fission. First of all, the movement will be stopped. All the other activities of the animal that will be stopped. This nuclei will divide into the two parts, that is karyogamy. Karyogamy means the division of the nucleus. And simultaneously there will be the division of cytoplasm that will be known as cytogamy. A fission is moving from one direction to another direction, and the animal cell is divided into the two parts. Two daughter nuclei which are formed, the genetic material will be distributed, and these two daughter cells, finally all the cytoplasm will also be divided into two parts. Initially they may have contractile vacuole or they may not have the contractile vacuole, and then the cytogamy is completed, the two daughter cells are formed, and each daughter cell is of a smaller size. When the food vacuole will be formed, the pseudopodia will be formed, and these two daughter cells will be converted into the amoeba. So this is how they are reproducing, which is known as binary fission, and this binary fission is always in favorable conditions. So we have studied these 4 slides. Number 1 was paramecium, number 2 was euglena, number 3 was amoeba, and this 4th one was when it is showing the stages of the reproduction, binary fission. If this 4th slide is placed for your study, then you need not to write the comments on the structure of the amoeba, because this is the slide which is showing the specific condition of this cytoplasm microscopic animal, which is the reproductive stage or the stage when it is dividing into two parts. So whenever the binary fissions stages are shown in the slide, you should not write comment about the detailed structure of the animal. Just you identify this is a slide of amoeba which is dividing, and this division is binary fission. Binary fission is in favorable conditions. Nuclei, division of nuclei is known as karyogamy. Longitudinal binary fission is there. Then division of the cytoplasm, that is cytogamy. Finally, two daughter cells are formed. Daughter cells are of the smaller size, and they are developing the remaining part inside the body, that is the food vacuole, pseudopodia, and will behave like an independent animal. So this is the 4th slide. So that is how we can write the comments about the various slides when you are going to the practical for the spotting. You should be very careful while drawing the well-leveled diagram, because if you will try to level all the parts of the animal, then it will not be possible for you, because the allotted time is very less. So be careful of selecting the characteristic feature of the animal, and the leveling should also be for the same parts. I hope by this method you will be successful when you will doing the spotting work, and you can get the good marks. In the next period, we will consider about the other slides of other. Thank you.